Okay, I'm Luc Ferrari, so I'm professor of toxicology in the School of Pharmacy in University of Lorraine. And so to be quick, we did mostly transcriptomics and cytotoxicity, and we share some data with other groups. We'll present some here. So our cells we use two models. We work mainly in vitro with submerged models, also some area do that later. So one model, our main model is NR8383, which is our macrophages from rats. And we also have a model of human macrophages, which are THP1 after some differentiation. So classically expose cells to nanomaterial for various time. For cytotoxicity, we use a 24 hours exposure, and then we use several techniques to assess IC50. This IC50 allows us to decide what concentration we'll use for the transcriptomic assays. So quickly, the transcriptomic protocol is rather classical. We treated the cells with not the IC50, but the quarter of the IC50 to have not a high toxicity, but to have the initiating events, to have the first answers of the cells to allow us to detect the more pathway as possible. So then the protocol is rather classical, binding to cyanine 3, to DNA chips, and then normalization of the data and data analysis, which were done using IPA with a choice of fold change, usually at 1.5, and p-value depending on the number of genes we want to analyze with IPA. So we also provided proteins to the group of Dublin for Hilary and David, and they identify the protein in the cell lysate and in the supernatants, and then we make some analysis also with IPA or string. So to go on to start with, I will just remind all of us that in fact there are some relationship between production of ROS and cytotoxicity, but this relationship is not for all nanomaterials. So there are some which produce ROS with a good relationship to concentration or to their functions, or to some other determinants, but not all. And cytotoxicity, it could also be, for some nanomaterials, related to inflammation in vivo. So this indicated that there are already some grouping of nanomaterials can be done. So we try to group not by cytotoxicity, but to group nanomaterials with pathway they initiated in the cells as an answer of the exposure of the cells to nanomaterials. And here, for instance, we compare two zinc oxides, the first one, to zinc ferric oxides. All cells have been treated with the quarter of IC50 for each nanomaterials, and we identified several pathways. And as you can see, we have pathways which are sometimes not regulated in both. For instance, mitochondrial dysfunction was not dysregulated almost, but oxidative phosphorylation was downregulated in zinc oxide and upregulated in zinc ferric oxide. For serotonin signaling, which is involved in inflammatory response and stress response, we can see that in fact both are upregulating, as well as EF2 signaling, which is related to endothelium reticulum stress, ER stress, which is upregulating. And we also get some interesting results in that case with zinc oxide, which is activation of cholesterol biosynthesis, probably due to large proliferation of the cells. We also try to compare not only the oxidation status of the nanomaterial, but also its form. And we did that with titanium dioxide, and we compare two forms, anatase versus rutile, and we compare both nanomaterials with the pathways they activated. And for the presentation, we decided just to take a fold change higher than 20. It gave us only a small amount of genes. Keep here all the 10 common genes, and you can see that we have mainly chemokine and proteins and genes involved in the inflammatory answer of the cells. So we also try to compare the different exposure we can have. As I said, we use usually with a submerged system. We also had access to our air-liquid interface, and we had the chance to collaborate with INRS, and mainly Laurent, Carole, and Laetitia, and we gathered the data for titanium dioxides. In each case, we use a concentration of 3 centimeters square per centimeter square for the exposure, and we check the transcriptomic data. So again, we have a lot of pathway activated, or genes activated, but we decided to restrain to a fold change of 1.5. And you can see here that, in fact, the common genes are rather limited. But if we check more strongly on the genes activated, we can see that for all of the exposure system, we have a chemokine activated, indicating that we have an inflammatory answer for the cells, and that is concomitant and is coherent with all the other genes we can have, even if it's not common to all. We have mainly inflammation. So we try then to compare different functionalization for the nanomaterials. For that, we have various carbon nanotubes, which have various functionalizations. So Sarah chose one gene without functionalization, one nanomaterial without functionalization, one nanomaterial with a carboxyl functionalization, and another one with an amine functionalization. We treated all the cells with, again, all the quarter of the IC50 and get the transcriptomic answers. So again, using a fold change of 1.5, as you can see, we have several genes activated, and you can see that in common we have only 48 genes, which are all related to the cytoskeleton, the ribosomes activity, and again, an inflammation answer of the cells. The difference, the genes which are not in common are different according to the status the functionalization of the carbon nanotube. For pristine genes, it was mainly an oxidative stress answer. For the carboxyl functionalization carbon nanotube, it was mainly genes involved in the traduction, so its production of proteins. And for the amine functionalization, we have mainly cytokine productions going again to inflammation process. So if we just consider what are the dysregulated pathway after all this exposition, we see that the pristine one has no pathway, quite no pathway in common with two activated, functionalizated carbon nanotubes. At contrario, the functionalization shows some common pathway activated for both amine and carboxyl carbon nanotubes. Again, it is mainly response to oxidative stress or inflammatory response or ER stress. So we have defense response of the cells. And we also have some different regulation, dysregulated genes, which are regulation of AF4 and mTOR signaling, so sometimes, again, protein synthesis. We also check some final endpoint, which is the cytokine expression in the supernatant of the cells. And as you can see on this slide, we have quite a similar answer for all the cells, which have each overexpression of cytokines and an overexpression for the amine compared to pristine carbon nanotubes, and quite, but it's not true for all cytokines, an underexpression for carboxyl. But in all cases, we have inflammatory cytokines expressed after carbon nanotube exposure to the cells. So we summarize this in this slide. We have in each case inflammation, but obviously the pathway which activated this inflammatory answer in the cells are not the same depending on the functionalization of the carbon nanotubes. So we also compare single wall carbon nanotubes and multi-wall carbon nanotubes to determine if we have different answer and different dysregulated pathway. We did that in both cellular models, rats, NAC8383, and the human THP1 macrophage models. And what was quite surprising is that we found that we have the same pathway dysregulated. But what was interesting is that for the single wall, what we have is mainly an activation of the signaling pathway. But for multi-wall carbon nanotubes, it was a decrease of this pathway, decrease of the activation. What was interesting is that, in fact, we have always we have quite the same activation between single wall and molecular multi-wall for in rats. And when we check in THP1, it was also the same kind of answer, but this time it was a regression for EIF2 signaling related to endothelial response, endothelial stress response. So again, we try to analyze if this activation and regulation of genes can have some signification at a protein level, and we analyze some the proteins in cell lysate and in cellular supernatant after 24 hours of exposure. And we had, again, a comparison between multiwall and single wall. And in cell lysate, what we found is an activation of lipid metabolism in both cellular model, and it was quite our only common point between all the data we have. But in some cases, we have some difference which appear. For instance, for the supernatant, we had between multiwall and single wall in rat cells, we have mainly difference, but not in THP1, the human cells, where single walls and multiwall both activated proteins related to exocytosis and the answer to virus, meaning the answer to some foreigners. This allows us to present some key events which are related to, which could be related to some adverse output, which can be similar or different according to the status of the carbon nanotubes or nanomaterials. In these slides, we present only molecular multiwall carbon nanotubes, but there are various in size, shape, length. Printex is carbon black. All others are carbon nanotubes, and the difference of size is related to the difference of K events, and at the end, probably it will be related to the difference in adverse output. I will finish with some return to the cytotoxicity data, because this cytotoxicity data can also allow some grouping of carbon nanotubes and express some difference according to the functionalization or their redox status. Here we have the effect of length on IC50, and cytotoxicity is higher when the length is under 500 nanometers, and the toxicity is less when we have a length from 1 to 5 micrometers. We have only one exception, which is here, which should have, because it's a 2 to 3 micrometer length carbon nanotubes, but in fact, it was established that these carbon nanotubes present more than 10% of impurities. So this allows us to also establish that purities of the carbon nanotube or nanomaterials is also a factor which is relevant to the toxicity of these materials. Hydroxylation has also an effect. We have here different couple of carbon nanotubes of similar size and shape, and the hydroxylation is only difference, and we see that when we have an hydroxylation of the carbon nanotubes, this will decrease the cytotoxicity. Carboxylation, in fact, increase cytotoxicity when we compare similar carbon nanotubes with similar size and shape. And the reduction, the redox status also have an impact. We have here 3 graphene oxide, similar size, similar shape, and we have different redox status. And it was clear that, in fact, when you increase the reduction power of the graphene oxide, then you will increase its cytotoxicity. So to summarize, we can, by all this data, propose several grouping to assess key events. Cytotoxicity is obviously a first grouping approach, but we can also check the several pathway and key events: inflammation, cholesterol synthesis, endothelial stress, endothelial reticulum stress. But we can also group nanomaterials by their functionalization status, mainly versus inflammation or DNA damage, but also nanomaterial size or length, grouped versus an inflammatory or genotoxicity answer. There are much more, but we have no time.